Thanks, Heather. Good to see you. Hi, everyone. It's a real pleasure to be here. I just want to thank Heather for all the hospitality over the last few days. It's been a lot of fun to catch up and to be in Philly. Yeah, definitely grateful to be here. And I'll give a talk going through some of our more recent research, but I'm also happy to talk more broadly afterwards about acupuncture research in general, about Society for Acupuncture Research. So you guys can definitely pepper me with whatever questions you want. So today I'll give a talk with some background on functional MRI, or fMRI, just to get everybody sort of up to speed on some of the techniques that we're using for some of our research. And then I'll talk about two different studies. One study is neuroimaging applications for carpal tunnel syndrome and acupuncture treatment of carpal tunnel syndrome. And another one is a more recent study that we've started working on just in the past year, looking at therapeutic alliance between clinicians, between acupuncturists and their patients. So first, a little bit of background on functional MRI and other imaging technology. So how do these things work, and how can we assess the brain activity in humans, in patients, non-invasively, right? So when you have neuronal activity, and let me just make sure, yeah, you see this. So when you have neuronal activity, that creates an electromagnetic response, which is picked up by techniques such as EEG, electroencephalography, and MEG, magnetoencephalography. But there's also a neurotransmitter response with excitatory and inhibitory neurotransmitters like glutamate and GABA, and even endorphin bindings that can be picked up by techniques such as PET, positron emission tomography, and HMRS, or magnetic resonance spectroscopy. And there's also a hemodynamic response. So when a part of the brain becomes active, we need to get the waste products out of that part of the brain. We need to get oxygen into that part of the brain. And so that's governed by the vascular response to the neuronal activity, increase in blood flow, blood volume, blood oxygenation. And that's handled by techniques like optical imaging and fMRI, or functional MRI. So this is done with an MRI scanner, but instead of scanning, say, for a joint capsule to look at ligaments and tendons and muscles, we're actually using a specific pulse sequence that is sensitive to the functional activity in that part of the brain. So you can think of this as, let's say these are neurons with blood vessels going around them somewhere in the brain. There's a basal state of neuronal activity. These neurons are firing with a basal level. There's a basal amount of blood flow and relative oxygenated and deoxygenated hemoglobin. And then when that part of the brain becomes active, there's an increase in blood flow to that area, an increase in oxygenated hemoglobin, and a relative decrease in deoxygenated hemoglobin. And it's this latter deoxygenated hemoglobin that leads to lower MRI field gradients around the vessels and an increase in MRI signal. So an increase in activation changes this ratio, which changes this parameter called T2 star and increases the fMRI signal. And what does this data look like? So basically, we have 40,000 little elements, voxels or volume elements inside of the brain. And there's a signal brightness in each one of those elements, and we can track that over time. And if we during an experiment, if we know what is happening over that time, such as, for example, let's say I put an acupuncture needle somewhere into the body, I leave it there in place, and then I stimulate over this period over here. This is called a block design, so I'm stimulating over here. Then I leave it in place again, I stimulate again, and then I look to see where in the brain does the time series of the fMRI signal brightness match what I'm actually doing with the needle. And so here you can see that every time I'm stimulating the needle over here, the signal drops. Then it rebounds back again, I stimulate, it drops again. And so what you have is these images where the colors, these pretty colors, are basically just coding the result of a statistical test. So when I match up the signal in that part of the brain with what I'm actually doing, and if it matches well, statistically significantly so, you can have either an activation or an increase in signal, which is over here, usually in red and yellow, or a decrease in signal, which is blue and cyan. Did you have a question?
About stimulating, are you talking about manual stimulation or electronic stimulation?
So this is just an example. I'm just trying to explain where this data is coming from, and then later on I'll talk about more about different specific experiments. But both have been done, and both can be done. So in our studies, when we're looking at acupuncture, I think it's important to look at different components of acupuncture. We know that with acupuncture, there's sort of the ritual aspect of it, the history, the palpation, the diagnosis, and the therapeutic alliance that's formed between the clinician and the patient. There's also then the needle itself and everything you're doing with the needle, such as inserting it, mechanically stimulating it, thermally stimulating it, or electrically stimulating it. And then the needles are left in place, and there's a relaxation response where the patient is lying on the table with the needles in place and is relaxing. And all of these things could potentially be important components of why acupuncture is working. Why is acupuncture relieving pain in patients? So in the first part of my talk, I'll talk about sort of this somatosensory element of the stimulation, and specifically mechanical and electrical stimulation and how this influences a part of the brain called the somatosensory cortex. So for this study, we've been working for a while looking at carpal tunnel syndrome, and carpal tunnel syndrome, or CTS, is the most common entrapment neuropathy. And so the entrapment is happening here within the carpal tunnel. This is the median nerve going through the carpal tunnel at the wrist. And what happens is really you have this vicious cycle of CTS. So there's an increase in pressure inside of the tunnel. That leads to ischemia and inflammation, which then damages the nerve microvasculature around the median nerve and leads to edema and fibrosis, which then fills in that space even more, leads to an increase of pressure even more inside of the carpal tunnel. And this basically produces pain and paresthesias, or kind of this numbness and tingling, to the fingers that are innervated by the median nerve, and that's the first, or the thumb, through the fourth digit. So the question is, how can acupuncture break this cycle? So one interesting thing about carpal tunnel syndrome, as opposed to some other chronic pain disorders, is that you have not just sort of these subjective measures in research where we're asking patients how bad is the pain, how bad is the tingling that you're feeling, but we also have these objective measures such as nerve conduction studies. And so here we place an electrode proximally to the carpal tunnel. We stimulate and we see how long it takes for this impulse to travel over the carpal tunnel, and pickups can be on the median or ulnar nerves. These are called nerve conduction studies. And basically mild CTS is the result of delayed sensory nerve conduction latencies and normal ulnar nerve. So the ulnar nerve is on the outside over here, so you can also measure the conduction of the ulnar nerve as well. And typically what you see with CTS is that there's a delay in this impulse because it's trying to get over this nerve, which ends up swelling and becoming like really fat. So moderate CTS is where you have these delayed sensory nerve conduction latencies and also delayed motor latencies, whereas severe carpal tunnel syndrome is all of these delays, plus you have reductions in motor amplitudes and even muscle wasting, which is evident over here in the thenar and hypothenar eminence of the hand. And this is a more serious medical condition, and these people we typically send off for surgery. So what we're focusing on here is how do we keep these mild and even moderate CTS patients from advancing more in their pathology and their disease to the point that they have to get the surgery. So one thing that we've done in our studies is look at not just the nerve conduction studies as an outcome, but we've also looked at brain mapping. And so with brain mapping, we're looking at something called somatotopy. And so here we know that this part of the brain, called the primary somatosensory cortex, this is it right here in red, has an organization to it that's reflective of the body, so it matches the body. And this was first discovered by a surgeon named Walter Penfield up in Montreal in the 1930s, where he had patients with epilepsy. He would open up their skull in order to attempt to treat the epilepsy, and the patients are awake throughout this process, and he would zap different parts of their brain and ask the patient where they felt that sensation. And by doing so in multiple patients, he was able to map out this organization of S1. And so you could see the head, the face over here, the hand, the torso, and down to the knee and the leg and the foot. And so you can see that this map is distorted. This is the common homunculus. And the distortion happens just because we're much more sensitive in some parts of our body, like our lips and our faces and our hands, than we are, say, in our back, right? So we need many more neurons or nerve cells in the brain to process all the signals that are coming from our lips and from our fingers than we do in our back, where we're much less sensitive. And so that's what leads to this distortion. So we're going to be focusing here on the part of the homunculus, the part of S1 that has the cortical representations of the hand. And so with functional MRI, we don't have to open up the skull, open up the head to try to map these things out. We can do this non-invasively in humans. And so this is, for example, an activation pattern with functional MRI for stimulation that's happening on the second finger or the second digit of the hand. and this is the third digit, and this is the fifth digit over here. And so you could see that as we go from the second to the third to the fifth digit, if you kind of plot the center of mass of these little activation spots, you could see it moving up the gyrus. And this is very similar to what Penfield found in his electrophysiological experiments, where as we move up the gyrus, we have the thumb, the index, the middle, the ring, and the little pinky finger. So the point here is that we can non-invasively assess these finger representations. We're getting some good light now. This is good. So what we found in our pilot study is that as opposed to healthy adults, where they have a nice separation of digit 2 and digit 3, and then up to digit 5 in S1, so this is the postcentral gyrus, or the primary somatosensory cortex, S1. In patients suffering from carpal tunnel syndrome, there's actually a contraction of the second to the third digit. So you can see these are much closer together than they are so in the healthy adults. Whereas the separation of the ulnar nerve innervated pinky or little finger, digit 5, is still nicely and normally separated in these patients. So that was a really interesting finding, and that was one of actually the first studies, imaging studies of carpal tunnel syndrome patients in general with functional MRI. So what we did then is that we sent these patients off for a protocol where they received acupuncture 13 times over the period of 5 weeks, 3 times a week for 3 weeks, 2 times a week for 2 weeks, and so on. And we used a combination of electrical stimulation, so low frequency 2 hertz electroacupuncture between PC7 and triple warmer 5. And then there was variable other points that were stimulated with manual acupuncture, and that was chosen by the acupuncturist based on the individual subject presentation. So there's this natural tension in research and acupuncture research between wanting to control everything and what's actually happening in the clinic with acupuncture, where you're trying to choose points that are individualized to each specific patient that's coming in. So our way of trying to work around this natural tension is to have a kind of this semi-individualized protocol where everybody was getting the electroacupuncture in these particular locations, but then other points were being chosen based on individual presentation. And this actually might be something that's easier to do for something like carpal tunnel syndrome than it is for like fibromyalgia or depression or other kinds of disorders. So that's point taken there. So what we found is that after a course of acupuncture therapy, so first of all, the healthy adults who did not receive acupuncture, this was just a test-retest comparison, still very nice separation between the second and the third digit. These are group maps, by the way, over multiple subjects. In the CTS patients improved in their pain and paresthesias. They also improved in their nerve conduction velocities or latencies. And there was also a greater separation between the second and the third digit in these subjects. So now we've gone from a pathological condition where there's contracted separation to one where we have much better separation in the second and the third finger in the brain. So our model for what's happening is that the pain and paresthesias in carpal tunnel syndrome are leading to more frequent and abnormal nerve traffic to the brain, which then through disinhibitory processes leads to wider, more extensive digit representations in S1. But it's not just more nerve traffic, it's also the fact that these paresthesias, this kind of tingling and numbness sensation that patients are feeling, are more simultaneous across multiple fingers. So normally, as we go about life and we touch things, we pick up a pen, there's going to be a delay, even millisecond delay between when our third finger touches that object and when our second finger touches that object. And it's this temporal delay that we have between our fingers that even leads to these representations in the brain. That's what creates that whole map, is that there's a temporal delay of when we touch different things. And what happens with paresthesias is that they're diffuse. They're not just one finger 1 minute, another finger another minute. They're diffuse. You feel them spread out over multiple fingers. And it's this simultaneity, so this is our contention, is that the simultaneity in signaling and this lack of separation temporally between different signals, that's what leads to, through something called Hebbian plasticity mechanisms, leads to less separated and more blurred digit representations or finger representations in S1. So that's what we think is happening here. And the question that we asked after this study is, can we link this neuroplasticity, this change in brain representations, with behavioral deficits in CTS patients. So, nobody comes into our clinic saying, hey, my digit 2 and digit 3 are too close to my brain. I can't, go about life. I mean, they come in because they're suffering from pain and paresthesias. So what is the link between the pain and the paresthesias and then what's happening in the brain with this remapping that's happening in S1? And what are the deficits that are resulting from this remapping? So for example, if you now have this blurring of the representations, and if I was to blindfold the patients and stimulate one of their fingers and ask them which finger were stimulated, would they be more apt to make mistakes because they have this blurring in their brain? So those are some of the questions that we wanted to ask. And so then we continued with another follow-up study, which with a much larger sample size. And here we had 8 weeks of acupuncture. We evaluated them both at baseline and at post-acupuncture periods. And then we had a follow-up, a clinical 3-month follow-up with something called BCTQ, which is the Boston Carpal Tunnel Syndrome questionnaire. That's just a validated questionnaire for the kinds of pain and paresthesias that the patients are feeling. So patients were randomized into 3 different groups. There was a local acupuncture group, which was fairly identical to what we did in the pilot study. Then there was a distal acupuncture group. And here we wanted to do real acupuncture, but not right at the site of the lesion. So the question is it important to be right at where the lesion is in the wrist, or can we still do acupuncture but have it be farther away? So here we did the acupuncture but on the opposite ankle. So if they had predominant right-sided carpal tunnel syndrome, we would direct the electroacupuncture to the left ankle. And then we had a sham acupuncture group, which was done with the Streiberger placebo, quote-unquote placebo needles, where the needle was not inserted, but we put them into non-acupuncture points, and we hooked them up to an electroacupuncture device that wasn't turned on, and so this was our sham control group. So one question is that people always ask is, why did you include this distal acupuncture group? Well, first of all, it's, we didn't just randomly choose this. It's consistent with mirroring styles of acupuncture, which many of you probably are familiar with in the room. So this is the idea that you can treat different parts of the body by mirroring them on the opposite limb on the other side of the body. So for example, if I wanted to treat the shoulder, I might do acupuncture at the opposite hip. And in this case, if I want to treat the hand or the wrist location over here, I would treat on the opposite limb at the ankle. So this is, I think there's Tan style and Tong style and different mirroring styles of acupuncture that actually apply this, as a real therapy. And so the other thing is we really wanted to control for local effects. We know that when we insert a needle anywhere into the body, there's going to be an increase in blood flow to that area, this axon reflex, and also some of the local effects that are happening there, such as the fascial remodeling. And so we wanted to control for that. We wanted to not have that at the site of the lesion, of the carpal tunnel lesion. Okay? So we we enrolled 80 patients with carpal tunnel syndrome, randomized 70 of them, and we did pretty well as far as dropouts. Very few subjects dropped out. We were able to retain 66 of them in the post-treatment evaluation, and even 55 of them we were able to get data on the 3-month follow-up. So from the standpoint of a clinical trial, that's pretty good. So what we found, first of all, is that we were able to replicate our findings from the pilot study in a different set of patients, a much larger set of patients. So this is data from 50 of those patients where we found contraction of the separation distance between the second and the third finger in the brain. And we were able to do this with both functional MRI and with magnetoencephalography. So again, a different technology also showing the same thing. So we think this is a pretty robust and reproducible finding. And the other thing we did is I alluded to this earlier, we set up a test where we had this device that we built with this vibrating little stimulators, and patients placed their hands over these stimulators, and we also had healthy controls, and they didn't know which finger was being stimulated, and we would ask them, or to report on a computer screen, which finger did they just feel being stimulated. And what we found is that compared to healthy controls, the patients made more mistakes for stimuli that were occurring on the median nerve innervated fingers, digit 2 and digit 3, and there was not a significant difference for the ulnar nerve innervated digit 5, which was a nice, interesting finding. But I think even more interesting was that the mistakes that they were making were correlated with the separation distance in the brain of D2, D3. So the more blurring they had, the closer these two representations in their brain, the more mistakes that these patients were making on this test. So this suggested that this somatotopy is not just sort of an epiphenomenon, it's actually related to behavioral deficits, and so it's indeed maladaptive. It's a maladaptive neuroplasticity that's happening in the brain. So something important to target, something important that we want to deal with in therapy. So the longitudinal results from this study, from this follow-up study, was that, first of all, we didn't find any statistically significant difference between the local and the distal acupuncture for the results of the subjective outcomes, the Boston Carpal Tunnel Syndrome questionnaire, the pain and the paresthesia severity. And so those two groups were merged together into one group for a lot of our analyses. And for the post-treatment follow-up, we found no difference between real acupuncture and sham acupuncture immediately after the therapy. They both were able to sort of successfully reduce the pain and paresthesias that the patients were feeling. However, when we looked at the long-term follow-up, the 3-month follow-up, whereas the patients that were randomized to the real acupuncture were retaining this improvement, those that were randomized to the sham acupuncture were reverting back to the pre-therapy condition. So we did find a significant difference there. And this is the data broken up between local and distal groups versus the sham group. So you can see here that both the local and the distal group kind of retain the results at the 3-month follow-up point. So the story was different for some of the objective outcomes, so such as nerve conduction latencies. This is where we're measuring the latency of the impulse over the wrist. And so here, with patients randomized to the Verum, the real acupuncture group, those patients were improving, so they were reducing the latency. So this is going in the direction we would want it to go in. We were reducing the nerve conduction latency, whereas those randomized to the sham group were actually getting slightly worse. The latency was actually increasing after the course of the therapy. And broken up into the local and the distal groups, there was a statistically significant difference for both of them relative to the sham group. So both of them were actually improving these objective neurophysiological findings, sort of the health of the median nerve at the wrist. Then we looked at in the brain and what was happening in the brain. And so again, similar to the pilot study, we found that with Verum acupuncture, there was an increase in this separation distance of these two fingers in the brain for real acupuncture and not for sham acupuncture. And there was a significant difference. Specifically, there's a sort of a larger effect size for the local acupuncture group, but the distal acupuncture group also was increasing this, but just not as much as the local acupuncture group. And the final results that we got here was that there was a predictive component between the separation distance. So those patients that improved the most right after the acupuncture therapy in the increase in the separation distance in their brain were those that retained their clinical benefit in the long-term follow-up. So there was an importance to remapping their brain. Those who remapped the brain the best were those who retained their clinical improvements in the long-term follow-up after 3 months. So our model is that while the local acupuncture might have effects both locally by changing, say, the blood flow parameters at the wrist itself, there's also potentially indirect effects through the brain. So the fact that we're stimulating, especially electroacupuncture, is then changing the afferents or the nerve traffic that's going up the nerve into the brain, and that could very well be working to remap these separation distances in this homunculus in the brain. And for distal acupuncture, the model there is that it's strictly this indirect effect, right? So we did see improvements with the distal acupuncture, so apply to the opposite ankle, and this has to be working through some sort of mechanism by which you're remapping the brain from the contralateral side to the ipsilateral side. So in the future, we want to look at the temporal order of this local and the brain plasticity to see what's changing first, the findings at the wrist versus the findings at the brain, or vice versa. And so this is kind of a representation of the model where you have this disordered, this irregular paresthesias nerve traffic that's happening along the median nerve, leading to the blurring of digit 2 and digit 3. And then when you have the electroacupuncture, what this sets up is kind of this larger regulated traffic that's going into the brain that's then maybe blocking out or swamping some of this paresthesia-induced irregularity. And it's this regular nerve traffic that then serves to then help to separate these different fingers, these median nerve innervated fingers in the brain. So we published these results in a journal called Brain, and we got very nice popular press coverage of this in the New York Times, in Boston Magazine. This was Time Magazine. In the UK, I love some of these crazy headlines. The weird way acupuncture helps carpal tunnel syndrome. It doesn't sound weird to me, but this is Le Figaro from France, picked this up too. We were on TV in Korea. Korean Broadcasting Service came out and filmed us. And we even got some fake news out of Russia. So it was fun to go through this. There was like a period of like a few months there where I was fielding a lot of journalism inquiries. But I think one thing that I also like to show is just how long of a process this was. So I graduated from acupuncture school in 2002, hired as a postdoc to really come in and start to do this clinical carpal tunnel syndrome research. So this really started in 2002, 2003. We were planning the study. We were executing the study in 2004, 2005, started working on the papers, the publications for this. Those came out in the next few years. Those were then leveraged to write for a grant from NIH, from NCAM or NCCIH now. And we were awarded this large grant in 2008 to do the follow-up study that was controlled with sham acupuncture and distal acupuncture. So that's what I just reported. We finally finished collecting all that data, writing up the papers. The paper was published just this year, in the spring of this year. And so you can see the long road. I kind of took you through that, but we're talking 15 years, right? So it's kind of daunting, for us in research. This is kind of what we do, but clinically speaking, it's like you want the results right away. You want to say, I have this like new idea. I want to study. I want to evaluate it, and I want the result to come out later that year. It just doesn't happen that way. So anyway, the take-home message here is that both Verum and sham acupuncture improve subjective and psychological outcomes such as BCTQ, while only Verum acupuncture improves objective or physiological outcomes such as nerve conduction latency and D2, D3 cortical separation. And so the importance here is that carpal tunnel syndrome is kind of unique, that we have these like objective outcomes. You look at low back pain, you look at fibromyalgia. These are chronic pain disorders, in some ways much more common than carpal tunnel syndrome, but we don't have objective outcomes for those things. We have to rely on self-report. We have to rely on asking patients, how bad is your pain? And so we saw what happened here with that outcome between Verum and sham acupuncture, and that might be leading to some of the equivocal results from clinical trials of acupuncture. So acupuncture, particularly electroacupuncture, is a somatosensory guided neuromodulatory therapy. I think there's been a lot of interest recently in what's called neuromodulation. This was a paper published in Nature, very prominent journal, about what are called electroceuticals. So this is coming. This is pharma coming in and is becoming interested in electrical stimulation to try to treat disorders. There's a lot of drugs that have failed, especially for chronic pain. Can electrical stimulation be used as a way to also treat these patients? And so they dubbed this term electroceuticals, but acupuncture, specifically electroacupuncture, could potentially be folded into these types of therapies. That could be a good thing, it could be a bad thing, right, for the field. So it's just something to keep an eye on, though, for all of us, I think. Local versus distal acupuncture may improve peripheral nerve function by distinct mechanisms within S1. And the question here is, could this be a basis for acupoint specificity? Could the homunculus and the primary somatosensory cortex in the brain be the rationale for why different locations in the body or different acupoints might be more or less effective for different disorders? This is just a speculation, but I think is very interesting. So I want to now transition from this study to looking at something different, and this is a study that we actually very recently started in our lab, just in the last year or two. And here we want to focus on not so much the somatosensory aspect of acupuncture and the needles, but therapeutic alliance. So this is kind of like the relationship between the clinician, the acupuncturist, and their patient, and what is the importance of that relationship. So we know that social interactions are actually quite important, and they shape how people feel, how people conceptualize pain, how people report the intensity of pain. So there was a recent RCT, in this case of spinal manipulation and exercise for chronic low back pain, a very large study, and this study found that patients' ratings of therapeutic alliance, which is basically their rapport with their clinician, was a major predictor of multiple pain outcomes and was a moderator effect. So this is an important component of all of our therapies. And the interaction style can be also experimentally controlled. So these are studies that were done in an irritable bowel syndrome RCT by Ted Kapchuk's group, where they actually controlled the interaction style. In some cases, the acupuncturist was communicating with the patient with an augmented style and going through a lot of different components, asking them about their life, asking them about their pain and how it impacts all kinds of different aspects of their health, versus a limited style where it's kind of like, just the facts, ma'am, and I'm just here to put in the needles, and there you go. And they found that the augmented style led to better clinical outcomes than did the limited style, for irritable bowel syndrome. And so the question is why? Why does that happen? I'm a mechanistic researcher, I'm a neuroimager. I'm interested to find out, why do they get these better outcomes? So our goal is really to understand the brain circuitry, supporting therapeutic alliance. What are the neural pathways by which empathy and therapeutic alliance influences pain perception? So why is this important to understand? Well, if we know how this works, if we know the mechanism, we could then go about augmenting it, right? Looking at different styles of interaction, looking at cost-effective trainings for clinicians as they interact with their patients. And ultimately, we want to reduce clinician burnout, right? This is a major problem right now. Our healthcare systems are changing through Obamacare, through everything else. And this is not just an issue for acupuncturists, this is an issue for all physicians, for clinicians. Burnout is becoming a major problem. So the question then is, how do we study this, right? So there are kind of social neuroscience research. There's people who focus on social neuroscience research. And the interesting thing, when we started looking into this, is that paradoxically, this has been typically studied with single individuals in isolation, right? So you're giving a stimulus to a clinician on a video screen, and you're just asking them, how do they respond to that stimulus? Well, that's very different from a social interaction, right? When we're talking face to face, that's very different than kind of interacting with text messages, for example. So understanding the brain circuitry, supporting these social interactions, we would argue requires two-person methods with an ecologically valid context. So one approach has been looking at two-person or even multi-person interactions is with something called hyperscanning. So hyperscanning really is the definition of that is the synchronous neuroimaging of more than one individual. So this has been done with EEG, I would say most commonly electroencephalography. And so this was a group in Italy that was looking at airplane pilots. So they had the chief pilot and the co-pilot. They would outfit them with EEG caps, and they had them take off in Rome, go up to altitude and then land again in Pisa, and they measured their brain concordance. How does the brain of the pilot and the co-pilot interact with one another, as they're going through these steps? And they found that there was concordance, especially during really difficult portions of the takeoff and the landing. So when they really had to work together, that's when the brain concordance was greatest. And so this, by the way, was in a simulator. This was not an actual flight, so don't worry if you're flying out in Italy and you have to go do this. This doesn't actually happen with Alitalia. So these methods have then been extended to look at juggling. So they looked at circus performers when they first started, and then as they were being trained to do juggling, and how their brains, the concordance would increase over time as they were being trained. And also with musicians, with duets, with quartets, they've outfitted, you could imagine like whole orchestras being outfitted with these EEG caps and looking at concordance, during the playing. So a lot of really interesting research that's happening in this field. Yes. So, when somebody, say, gamma band increases, does another person's gamma band increase at the same time if they're interacting in a social manner? Things like that. So the question then is, what about functional MRI, right? We want to look at also if there's concordance with functional MRI, which is not just a cap being placed on there, but you have to have people going into an MRI scanner, right? And there's reasons you want to do this. There's better spatial resolution with fMRI than these EEG methods. We can also look at deeper in the brain. Is there concordance in subcortical structures? This is something you can't really get at well with EEG. So we wanted to look at functional MRI hyperscanning, which is technically quite difficult. People have attempted this. This was an attempt out of Princeton where they actually built a coil that had two different components, and you stick two people in this coil, but you can't really do this with clinicians and patients very well. So this was not a good solution. So we wanted to look at ways to actually use two MRI scanners at one time. So we had two separate synchronized fMRI scanners, and the question we asked is, how is patient-clinician brain concordance associated with therapeutic alliance? And how does it impact analgesia? And what brain regions demonstrate this concordance? So our hypothesis is that the degree to which clinicians mirror neuron systems accurately reflect the patient's own brain responses to clinically relevant stimuli plays a critical role in nonverbal cues and the patient's perceptions of therapeutic alliance with the clinician, and then ultimately that can lead to improved clinical efficacy. So we set up basically these two scanners. We got some MRI-compatible video cameras and audio microphones that we'd set up into the scanners. The two MRIs were then synchronized with an external trigger. So we had basically a laptop that was sending out a signal to each of the scanners, and the scanners could then be started synchronously, through this connection with the laptop. And we were also able to track the network delay because all this was happening over a local network. It wasn't hard cabled. And so, you've probably experienced it when you're using Skype and you're talking to somebody far away, and then suddenly, it becomes kind of, when you get to more than a few hundred millisecond delay, you can really notice, and it doesn't feel like a natural interaction. So we wanted to avoid that. So we are keeping our network delay. We tried different ways of doing this, and we were able to get our network delay down to like tens of milliseconds or even less than 10 milliseconds. So that's pretty good. So we have the patient in one scanner, the clinician or the acupuncturist in the other scanner. Inside of the patient MRI room, we induce a pain stimulus with a cuff. We do this in a lot of our other studies with chronic pain patients. So we give them a pain stimulus to see how their brain responds to that pain stimulus. The patient then can use a button box, which they're holding to rate the pain that they're feeling. And for the acupuncturist, the needles are inserted right above the cuff over here. And the acupuncturist then has their own button box. Then they go into their own scanner, they get their own button box, and they can use different buttons to then affect an electroacupuncture stimulation. So when they press a certain button, electroacupuncture gets delivered to the needles of the patient in the other room, and we can use this to reduce the pain that the patient is feeling. So we're collecting cardiac, respiration, and skin conductance data in addition to the bold fMRI data. And the way the experiment is set up is we have two different conditions. One where there is an interaction between the clinician and the patient. So they come in, they do an intake, they take the pulse, they look at the tongue, they do whatever it is that they want to do. And we leave it up to the acupuncturist to decide how they want to do that intake. Then they go off into their different scanners, and they have this interaction. And I'll tell you about that in a second, with MRI. And then they come back again and they see a different patient, and this time they don't have an interaction beforehand. They just go into their scanners, and it's just kind of like this unknown person on the video camera that they see in front of them, but they didn't have a previous relationship built up with them before. And the same thing goes for the clinicians. So it's kind of like randomized, it's paired randomized. So each clinician sees two patients, and each patient sees two clinicians. So during the scan itself, there are cues where both the patient and the clinician see whether during the next pain stimulus they will be treating or not treating, and the patient sees whether they will be treated or they will not be treated. And then we have three different conditions. For the no treated, they just aren't treated. For the treated, they're either treated with real acupuncture or sham acupuncture, where the current actually doesn't flow into the needles. And then they rate the amount of pain and kind of the feelings that they had about the procedure in the previous block. And so what we found is that during the treatment, there's a reduction in the pain that's being rated by the patient compared to the no treatment condition. And that was also, we also asked the acupuncturists, how much pain did you think the patient was in? So the acupuncturist sees the patient, they see a video of their patient when they're receiving this pain. So, based on whatever way they want to do it, their face, anything else, they are also rating how much pain they thought the patient was in during that last stimulus. So this relationship between the treated and the no treated condition, and for this particular experiment, we can interpret as the placebo effect. There was actually no difference between the real and the sham EA condition for this particular case, and that was partially by design because we really wanted to focus this study on the relationship itself, the therapeutic relationship, and less so on some of the somatosensory elements of the acupuncture. And so what we found is that, first of all, this is like really striking to me. This is the patient's brain response to feeling the pain, right? So this is they're feeling the pain. You see activation in posterior insula and some of these areas over here, but you also see activation in, this is called the temporoparietal junction, and this part of the brain is important for what's called theory of mind. So when you're in a cognitive task, when you're asked to imagine yourself in somebody else's shoes, in somebody else's body, and what does it feel like, when they're going through whatever they're going through, that's called theory of mind. And that's been localized to several brain regions, including this temporoparietal junction. This is the brain response in the acupuncturists, in the clinicians. It's huge. It's like the clinicians are not experiencing any pain. They're just watching their patient experience the pain. And the activation that they're seeing in their brains is in some ways looks much more robust than the patients who are actually experiencing the pain themselves. I thought that was very interesting. But if we look at what part of the brains are activated in both the clinicians and the patients, that goes down to these areas over here in green. And this is the TPJ, the temporoparietal junction, and the ventrolateral prefrontal cortex, which are known to be really important for social mirroring. So there's these mirror neurons that we have in our brains that activate when we see somebody going through something. And there's mirror neurons for motor, for activity, such as grasping. There's mirror neurons for somatosensory, so tactile, feeling, touch. And there's also mirror neurons for empathy, for kind of social relationships. And these are the social mirror neuron systems in the TPJ and in the ventrolateral prefrontal cortex. So this is very nice. We're actually able to identify these areas with functional MRI. And so what we found is that there's greater brain concordance or co-activation in these areas during the treated condition versus the no treated condition. And there was a relationship between the amount of concordance and the analgesia that's reported by the patients. So if there's more concordance in these areas, the patients are reporting a greater reduction in the pain during the treated condition than the non-treated condition. And so this is still kind of pilot. None of this is published yet, but we're hoping that these results stay solid, as we're getting more and more experience and scanning more and more individuals or dyads. So the other interesting thing we're doing is that because we have these videos, we are tracking the expressions that both the patients and the clinicians are showing. So there's new tools now that are out there that have been used for artificial intelligence and machine learning to try to track what people's faces look like during different conditions, right? I mean, you could see a lot of commercial applications for this. You're going through the supermarket and you pick up a certain product. Wouldn't it be great to know whether you're smiling when you pick up that product or not, or when you're tasting that product, So you can see a lot of applications for this. It's frankly a little bit scary to me. But these tools are out there, and so we're using that to look at the patients and the clinicians' faces when they're experiencing them, when they're showing emotions in response to their stimuli. And basically what happens is you take different expressions, sort of these baseline expressions such as nose wrinkling and upper lip raise, and these are then mapped into canonical emotions such as joy, anger, disgust, surprise, fear, sadness, for example. And so you're mapping these expressions to these kind of canonical emotions. And what we found is that it was the emotion of disgust was the closest emotion we could find to pain. So if you notice, there's no canonical emotion for pain, so we needed to do this mapping on our own. And what we found is that during our experiment, when we gave a stronger pain stimulus, so this is P40 or 40 out of 100 pain versus a non-painful squeeze of that cuff that I showed you before, this emotion was significantly increased in the patients, right? And this was the case also for the cue. This is when they saw the cue of what kind of pain they were about to receive. We also saw an increase there, and this was the only emotion that we saw this for. So this is kind of the emotion that we're using to track the patient's response and their facial responses. And so what we found is that, and I think this is really interesting, there was no relationship for the patients between the emotion, the amount of disgust that they were showing on their faces, and what they were then eventually rating as how painful that pain was, right? Because you can imagine there's a lot of different people with different personalities, and some people show their pain, some people don't show their pain. That's not going to get picked up in this. But for the clinicians, we did find a significant relationship. So the more disgust the patient was showing, the greater the pain the clinician thought the patient was in. So I think this is really interesting data. And what we eventually want to do is tie in these vicarious pain ratings from the clinicians and the amount of disgust that the patient is showing with analgesia, with therapeutic alliance, and some of these other parameters that we're also tracking in these dyads that we're scanning with the hyperscanning. So the conclusions here is that therapeutic alliance can significantly shape clinical outcomes. Understanding the brain circuitry supporting social interactions requires two-person methods such as hyperscanning. And the concordance between activity in clinicians' mirror neuron systems and patients' brain response to clinically relevant stimuli might support nonverbal cues and patients' perceptions of therapeutic alliance with the clinician, leading to clinical outcomes. So this study is ongoing. I think we've scanned 23 dyads. We hope to get to 40 by next summer, and we hope to publish after that. Sure. This is all being recorded, by the way, so you'll probably have access to it through the association. So if you're not a member, become a member. But in any case, this is the end of the talk, and I just definitely wanted to acknowledge my lab, because really this is a product of everybody that's been working hard in the lab. For the carpal tunnel work, a lot of that was done by a postdoc named Yumi Maeda, who has since gone on for a dentistry degree. And the hyperscanning studies that are ongoing are being supported, really run by Dan Michael Ellingson, and who's a postdoc here from Norway, and many of the research assistants that are also contributed to this. Ted Kapchuk is a co-PI with me on the hyperscanning studies. Joe Adet worked a lot on the carpal tunnel studies that we had ongoing. So just wanted to acknowledge all their support, and the support of the Korean Institute of Oriental Medicine, who I have a close relationship with in Daejeon, Korea. And they've sent over some folks for training in my lab, and I go back and forth and we work with them closely. And of course the support from the NIH, who has given us grants to do a lot of this work. So thanks very much. And thank you. And if I can put on my SAR hat for a second, I definitely wanted to give a plug for the Society for Acupuncture Research. We are a loose band of academics who put together these acupuncture research conferences that are international pretty much every other year. We get a lot of attendees, not just from the United States, but from Europe, from China, from Korea, Japan. And the next one will be in 2019 in Burlington, Vermont. So, and I think it should be very, very relevant to clinicians. We're trying to figure out ways to get more clinicians to come to our conferences. And so I'm very happy to get some sort of feedback as far as what kind of content would entice you guys to become members in SAR and to come out to our conferences. So thanks a lot. And if you guys have any questions, I'm very happy to answer. Yep.
Great job, by the way. Fantastic. So in your CTS study, did the study results correlate with clinical outcomes and perhaps any like neuroconnection studies or EMG results? So your outcomes showed the neuroimaging results, right?
Right.
Did you happen to correlate it with how the patient, did the patient have less symptoms and were there any neuroconnection?
Right. So the relationship that we did find was the predictive relationship that I showed. That the remapping in the brain was then, actually it wasn't just a predictive, it was the remapping in the brain was correlated with the clinical outcomes, the reduction in paresthesias and pain severity after the therapy, but also was predictive of long-term outcomes. So that was the relationship that we found there. Yeah.
How was it that we increased our brain concordance?
That's a great question. This is, I guess I should say, there's maybe 4 groups in the world who are doing hyperscanning fMRI. None of them are doing it on clinical context. It's like joint attention and cognitive tasks and stuff like that. So we're at the very beginning of this type of research as applied for clinical outcomes, much less anything having to do with acupuncture. And so it's just very early days. So right now we just wanted to see whether there even is greater brain concordance that's there with clinical outcomes, and if that's related to clinical outcomes. If we can show that, then we can try different interventions, different training styles that then heighten concordance more efficiently than others. And the interesting thing is that it might not be strictly a time thing. You talk to hospital administrators and they say, well, this is all great and good, but we can't, Dr. X doesn't have an hour to spend with the patient, for this. But it might not be something like time. Time might not be the most important variable. It might be something that you actually develop just in the first few minutes with a patient
That
then carries over.
Eye contact or posture, open heart posture, or what are
Yeah, so we're So yeah, exactly, perfect. So we're recording these intakes. The intakes that the acupuncturist has with the patient, those are being recorded and we're scoring them. We're going through them, we're monitoring how much these different things are happening, and we're hoping then to relate that with some of the imaging outcomes that we get from the imaging studies. So we're hoping to do that, but it's difficult. It's difficult to do that. Yep.
Are you doing, or are there other people doing recording studies with interactions that are not necessarily clinical, but could have almost a similar mapping of like religious figure with their congregation, with any other sort of thing that could also interfere?
Yeah, there's a lot of people who have sort of come out of the woodwork, wanting me to like extend this to there's a guy, and I'm not talking about you necessarily, that might be a great idea, but I had there was a neurologist who's studying frontotemporal dementia and these patients as they decline and how their relationship with their caregiver, their wife or their husband, for example. And he wants to put that sort of dyad into the scanner to see, what happens, because it's a very difficult relationship, when your partner is declining like that. And they want to do some of these kinds of studies to look at that relationship to see if there's a break in brain concordance that's happening, over time in these people. And that, I've had a lot of, autism, a lot of applications for autism, as you might imagine. So there's a lot of interesting directions that this kind of research can go in, and we just got this like up and running.
How hard is it to even just get 2 fMRIs like back to back so you can actually pull this off?
Yeah, so we're at an large imaging center. We have 8 human MRI machines there, and it's still not easy. It's very difficult to book these things because we're jockeying with a lot of other groups that are doing other research in science and finding 2 scanners at the same time. And then, you think of no-shows, right? You have like 1 scanner and the patient no-shows, well, that kind of sucks. But now you have 2 scanners. Now you've doubled your chance of a no-show, be it the patient or the acupuncturist who doesn't come. It's just, it can be maddening trying to get these things done.
Can I ask one more on CCS? Was there a reason you didn't bring the fMRI back in at the 3-month follow-up?
Cost. It's expensive to do this, and you kind of limit. But then maybe the next generation of the study will think about doing that. Yeah.
I feel like I missed, there was a slide where you had compared local versus distal treatment at the 3-month follow-up. I think you said there was no difference, but I thought the graph looked somewhat different. Can you talk about that?
Yeah, so that might be a statistical difference. So for some of the comparisons, we might be underpowered. So you might see what's like an effect size difference. So you look at the graph, you see the bar plot, and it's larger, say, for local, smaller for distal, but it's not statistically different, and that just means maybe if we had 100 more patients, we would see a statistical difference, but we just didn't in that case.
And the last one. Any percentage come to your minds about placebo effect? Like after all is said and done, what percentage of the effects seem to be the placebo effect?
It could be a whole long discussion just to talk about that. I have problems with first of all, the pejorative connotation of a placebo effect and what that means. We do acupuncture. We don't inject anything with our needles typically. And so we try to take advantage of endogenous healing mechanisms. What is a placebo? A placebo is an endogenous healing mechanism. It's a way that the patient either gets better or reports feeling better that is happening from an endogenous mechanism. So I just think because the way placebo effects have been thought of because of pharma research, because of drug, and with drugs it's very easy to develop like a sugar pill, and you have that as like a control. It's much more difficult to do that for devices. It's not just acupuncture, it's any device. It's surgeries, doing sham surgeries. There's actually been multiple clinical trials of sham surgeries that have shown that sham surgeries are quite effective. It's very effective, right? Where you just open up the knee and you don't do anything and you close it back up. Those people get just as good as debridement in knee surgeries. So I guess the question is, I think it's a wrong question to ask, well, what is the placebo component of acupuncture, or how much are people getting better in these sham trials? There could be many biological mechanisms by which people are getting better in the sham arm of a clinical trial that are not necessarily like placebo effects per se. Yeah. Which is why we're interested in therapeutic alliance and looking at these kinds of relationships.
Yeah.
I don't know if that answers the question, but that's something that I think of when I read some research articles on whatever, with local acupuncture versus distal versus sham, because I've spent some time in the past decade studying the Napadow acupuncture and Richard Tan stuff. And there is no, I mean, there's meridians and all this stuff that we're taught to understand, but really anywhere there's a life force, there's chi, and anywhere you put a needle in, you're stimulating that force. So how do all these sham acupuncture studies, or studies that use sham acupuncture, how do we make sense of what they do and don't find? And because I learned points in studying Napadow acupuncture that aren't on any meridians.
Right.
And that's, you know, like I have a real, like I haven't
Well,
figured out how to
yeah.
understand that.
So, I mean, there are different ways that sham acupuncture has been done in clinical trials. So you're right, there are some studies where you actually insert a real needle into the body, but at a non-acupoint, and that could very well be a tongue point, for example. Then there's other studies where the needle is not inserted, right? And it's not inserted either on an acupoint or not on an acupoint. And so there was a recent meta-analysis that was done looking at a whole consortium of clinical trials that found that those where the needle is being inserted into the body get better benefits than those, I'm talking about just the sham group, the placebo group. Those get better benefits than if the needle is not inserted. So it's a sham needle that's not actually inserted into the body.
So it goes to radiation.
Exactly. It's called a striker needle.
Well then, and I haven't studied Toyohari, but then you can bring them into the whole.
Right. And so there might be biological effects of actually tapping on the skin that's happening from those. I mean, I don't know if you've ever seen a Stryper needle, but those needles, there's a lot of heterogeneity. I've even seen those things draw blood before. I mean, because they're like tapped down on the skin and they can scratch, because it's just like a dull needle that then contacts the skin. So there's a lot of heterogeneity there too. So it's hard to know what's going on. That's why it's hard to interpret those data because of it. Yeah. Great. Well, thank you very much.